What are fold switching proteins? So fold switching proteins are these very interesting class of proteins when you have the same primary amino acid sequences adopting two different conformations. To give you an example, this is a transcription factor, RFAH, that binds to RNA and DNA polymerase, and it undergoes a change from alpha to beta in its secondary structural conformation, as you see in the C-terminal domain, which binds to its partner. And why is this important? Because this particular phenomenon questions the one sequence, one fold paradigm. And also, I want to impress upon this, and I will get back to this point many times during the presentation as well. These are evolutionarily selected for and have functional and regulatory roles. So again, to emphasize on the importance of fold switching is crucial because it plays an important role in regulation, and they are also present and they have effects on different human diseases, and they are widespread mechanism largely undiscovered in many biological pathways, as we see here in these examples, where you see how the switches happen and where they are implicated. But again, the point that I want to make in this presentation is that fold switching is an evolution adaptation rather than artifacts or isolated functional changes. This is what I want to impress upon you. Let's talk about a family that actually adopts two different folds. So this family of bacterial response regulators, if you look at the N-terminal receiver domain, that aligns very well structurally. But when you look at the C-terminal DNA binding domain of this family, they have two subfamilies which have two different folds. One has a beta linker and a winged helix, and the other one has a helix linker and a helix turn helix DNA binding domain. So the question comes, how does one family evolve these two different folds? So mainly people have looked at how new protein folds evolve by mixing and matching of stable scaffolds of super secondary structural elements. But there's another way that it could lead to two different protein folds. That could be evolved fold switching or stepwise mutation that lead to two different folds. And most likely, this is what is happening in this family. As we look at more of the data, it will become more clear. So how do one go about to identify such mutation-induced fold switching? So for this purpose, what we needed to do was to find pairs of sequences which have moderate sequence similarity but differing secondary structure, which we can see in a very nice example here. So one takes the full length sequences, compares it with all PDB sequences that are available, performs alignment, and then you see that there is moderate sequence identity, which means homology. But at the same time, you have these alpha helices that align so well with beta sheets. So this could be a possible example of mutation-induced fold switching. If it were not to happen, you would have identical secondary structures here. So again, what we did, so I'll explain two steps of this process. The first step was to find pairs with moderate sequence similarity but different secondary structures. And we did this by conducting 3 rounds of position iterative blast on fixed PDB sequences, and we received good scores, a very nice alignment coverage, and significant e-value, which made sense of our search. And what we saw, 11 of the top 20 hits shared the winged helix fold, which shared its topology with the KTPE subfamily, while 9 had the fold of fixed A. So that tells us this alignment contains both these two different domains. So we also performed a reciprocal SciBlast search using sequences of KDPE, PDB, and searched through the sequences of fixed J. And what we found was good sequence alignment and coverage and an e-value of significance. And we could see that we could match these sequences with the other fold as well. And when we looked at the alignment, we found that the C-terminal domain, which aligns very well sequentially, and when we look at the structures there, they are different. So let's talk about the second step of it. So we did PsyBlast, but we wanted to make sure that what we are seeing makes sense. So we went on to do a more sensitive search using Jackhammer, which uses iterative hidden Markov based alignment, and to make sure that it's more sensitive and picks up even more sequences. So what we did was that we took 23 non-redundant full-length response regulators with 11 of them marked as helix turn helix and 12 with winged helix. These sequences were then searched against a non-redundant database of protein sequences of salt structures which had 70 helix turn helix and 95 winged helix proteins. And this alignment was done using Jackhammer to make sure that we have a sensitive search. And we looked at the e-value and the distribution and the sequence alignment. What I want you to focus on is this figure, where you see there's very nice clustering of all the sequences of helix turn helix, and these are the sequences of winged helix. So they cluster very nicely. And another thing I want you to follow is this yellow region, which is the linker. So N-terminal domain, the sequences are aligned really well, the structures are aligned very well. Whereas for the linker, you see there are some insertions and gaps there. And C-terminal domain, you really see how the helix changes to wing domain here. So that tells you that this is a possible example of evolved fold switching. So with that, what we needed to do was to really curate sequences for subsequent phylogenetic analysis. So now that we know these two uh, subfolds exist in this protein family, we want to understand how they came about. And to do that, we have to make sure of the sequences that we start with of our data set. So we took all our sequences of the fixed shape sequences of salt structures from PDB, and same with KDPE. And these were queried against a non-redundant database using Protein Blast. And we identified around 600,000 putative homologs, which is a lot. So what we had to do was really form clusters of uh, of similar sequences and see if we can filter them down. And we used a greedy clustering algorithm just to break them into clusters. We had about 400 clusters, and then we filtered them to 85% redundancy for a final number of 23,000 sequences. And these sequences, when you looked at them, there were around 11,000 KDPE-like sequences and 13,000 fixed J-like sequences. That's quite a lot. But I also wanted to talk about bridge sequences here. So these two families, when we looked at them and we wanted to bridge them to form a big MSA, multiple sequence alignment, we found it was hard to do so. What we needed was some sequence to bridge these two subfamilies together. And what we did was like we organized our sequence and looked at them in terms of sequence identity to find if there was a point where it switched to the second fold. In terms of sequence identity, TME 68356 was an interesting protein because this clusters with the KDPE, but it's annotated as a uh, helix turn helix. So that makes it interesting that it seems to have characteristics of both families. So using these, we may be able to bridge the gap and put the two subfamilies together. And bridge sequences have often been found of adopting different conformations. They are also fold switching sometimes. So they are interesting to have and to annotate in the family. So using that, we built a MSA. This is an interesting depiction of my data, and I wanted to share it because it's important to understand that having a lot of data is great, but a lot of data also means a lot of noise. So when you have a multiple sequence alignment, which is composed of 23,000 sequences, and they have enough identity, but also they are dissimilar, you will have gaps, and these gaps will lead to a very noisy tree, which you cannot get much information for. So, what do we need to do? We really need to whittle it down to a set of sequences that are unique enough but also share characteristics of both of these subfamilies. So for that, we perform something called cross-family sequence searches, which we also did earlier when we were trying to build our data set, when we were searching KDP-like sequences using fixed J-like sequences. So what we did was, let's just follow the figure because it's easier to follow there. So find all the sequences that are homologous to fold 1, and I've kept the color scheme same everywhere to, you know, for us to remember. So this is fixed J-like, and this is KDPE-like domains, right? So these are the sequences that belong to fold 1. These are the sequences that belong to fold 2. We cluster them, and then we compare them. We do cross-family searches to find enough sequences that represent both from these clusters and from these clusters. And after obtaining a cross-family sequence set and then performing an alignment, we had an MSA which consisted of around 3,000 unique sequences, which had 664 KDPE-like and around 2,000 fixed J-like protein sequences, which made sense. And from there, we could move on to form a phylogenetic tree. And we needed to do this because we wanted to explore the evolutionary relationship between these two folds. And we did that by estimating the maximum likelihood tree using fast tree. So here again, we see the sequences are very nicely clustered between winged helix and helix-turn helix. And this tells us that these two subdomains or subfamily have a common ancestor. But we cannot really tell where the bridge sequences actually lie. Here it looks like they actually lie in between the two uh, clades, but we are not sure because this tree is not rooted, and we cannot comment on whether um, the winged helix came before, or the helix turn helix came before. So how the path of evolution has occurred. I I'm just realizing I've been talking a lot. Do you have any questions that you would like me to answer and go over some of the data before I move on ahead? I'm guessing I'm good. So I'm going to go on ahead. Right. Before rooting the tree, we wanted to make sure that the evolutionary relationships that we see in the tree, they make sense and they are validated. For that, we performed bootstrapping support and build a consensus tree. So bootstrapping values in a phylogenetic tree really tells you how many times the same branch is observed in the tree when repeated over a generation of time. So we did it for uh, 1,000 steps of bootstrapping, and it gives you a value of 100. So the better support you have, more towards 100, that particular connection or branch is well supported. So this is how we colored it here to just show you that there are certain things which we are not sure about. Mostly we are happy with the tree. Now that we are happy with the tree and we are happy with the way things look, we wanted to root the tree. So, how did we go about it? So, rooting the tree can be done usually as, you know, something assigning a special node as the tree on the tree, representing the most recent common ancestors of all the taxa in the tree. So, usually people do uh, in two ways. They would either choose an outgroup. Outgroup is a sequence from the set which is distance enough but also close enough to the other sequences, so you can safely say this is something which should be where it should be close enough but far enough from the tree when one can see the point of evolution. But for us, it was more difficult to find it because they looked. close enough to everything from our unique set. So we decided to infer rooted trees without an outgroup. So people basically use non-reversible model or Bayesian model. We use non-reversible models, and what we did essentially was a very brute force way of assuming that each node is a root, and then rooted the tree at each particular node, and we generated close to 6,000 trees, rooted trees, and then we analyzed them using scores which are represented by p-values, an unbiased AU test, which gives us a p-value. These are the around 6,000 trees that we built. I just wanted to show you the distribution of all the p-values here. And what really tells us is there were a few of them which had p-values greater than zero point 0.8, which just means that those trees are most likely to be correct. The root was placed at the right position. So there were 18 of such trees, and we looked at all 18 of these trees. So what were we trying to answer using this method? We wanted to see where the bridge sequences lie, and we wanted to see, can we understand whether helix turn helix came first and then evolved into wings turn helix, right? So these were the two questions we were trying to answer with all the calculations that we did. I wanted to focus on that as well because it might get lost in the weed. Right. So these are the two trees that I'm showing. We looked at all 18 of them. And what we can see is that it seems like most likely the um helix turn helix came first and gave rise to the winged helices. And here we have the bridge sequences. I'm just showing one here, but all the bridge sequences were mostly at the interface between the two. Same here. So, what does this tell us and what can we do from here? So, now that we understand that the bridge sequences can help us understand how the evolution of these two sub families occurred, and we have an idea that fixed J uh, helix turn helix came before winged helix, we wanted to reconstruct the ancestors or do ancestral sequence reconstruction to actually see how this mutation of few residues lead to differences in two different folds, right? How they give rise to these two different folds. So again, using the same ideas, we estimated the ancestor sequences for each of the nodes, and we collected sequences of ancestors by calculating the probability of each of the 20 amino acid in each position. So for each of these ancestors, we calculated whether that particular node can be an ancestor and what could be the sequence of that ancestor. And we generated again a set of 6,000 ancestors. And after looking at each of them, since we know that bridge sequences are important, we wanted to find out 5 ancestors which were closest to the bridge sequences. So we found out the sequence identities of these ancestors which are closest to the bridge sequence, and that's how we could whittle down our list to 6 different ancestors closest to our bridge sequences. And for each of these ancestors, posterior probabilities or each amino acid states at each node was calculated again. And what I would like to show you next is using these ancestral sequences, we gave these sequences to AlphaFold and we predicted the protein. And I'm just showing you the a part of the linker region and the C-terminal binding domain, because that is where you see the switch happening. So this is your bridge sequence. These are all the ancestors, and this is where all the winged helix and the helix turn helix lay. What I would and the numbers here, this is the probability of that ancestor being most likely correct, and this is the sequence identity of the ancestor to the bridge sequence. What I would like you to do is just follow this pink helix here. So as you see, the pink helix and this yellow sheet here. As you see, as you move down the tree and as the sequence changes, when you move towards winged helix, how these change to beta sheets. And as you move towards helix turn helix, and you see these two regions change into helices. So you can really see a pathway of mutation that leads to these two different folds. And also here we have the bridge sequence which we identified before. So this really tells you how this occurs. What we are trying to do is select some of these ancestors and express them and actually perform experiments like look at CD and maybe some form of NMR to see if we can actually see this change happening in experiments as well. But we have seen that through prediction using AlphaFold. So this really gives you that idea of how mutations change fold in a protein family and give rise to these two different clades, right? But are we the only ones talking about this? We'll get to that. But I also wanted to talk a little bit about functional aspect of this. So have these folds, the winged helix folds, evolved to have more specific contacts with the DNA? And for that, to understand that, what we did was we looked at all the salt structures of helix turn helix, which have contacts with 17 unique nucleotides, and winged helix contact around 22 unique nucleotides, which just tells you that there is more specific contacts for winged helix versus helix turn helix. And these are just representative examples showing how the specific contacts change. So there's a there's a functional reason for this adaptation as well. But coming back to the question, is this something that we have observed in this particular family? What have other groups seen? And how does fold switching occurs in other protein families? Um, so I will talk about that in a few slides. I'm jumping the gun here, but here again, the important takeaway is that evolved fold switching could explain the presence of two different protein folds in a protein family. And by performing phylogenetic analysis, ancestral reconstruction, we could provide insight of how this process takes place when you have few mutations that changed helix turn helix to winged helix, and this could have been uh, evolved to provide more specific contacts with the DNA. But I really want to talk about fold switching that happens in other families. So this one paper, this came out in Science 2021. So this talks about the human chemokine XCL1 or lymphotactin fold. This is very interesting because this fold switching occurs at equilibrium. So the chemokine fold is monomeric and the alternate fold forms dimer. So this is a dimeric fold. And this group also performed ancestral reconstruction, phylogenetic analysis, and they also followed up with experiments to look at how one fold, the leading from the chemokine fold, has led to uh, two folds, and they have pointed at a branch where there is a loss of conserved disulfide linkage that and also with accumulation of mutations, additional mutation that lead to the XCL1, which actually uh, can exist in two folds at equilibrium. So yeah, this was a very interesting paper that also talks about fold switching and how it happens in this family. In our lab, we also are interested in RFAH, and I said this protein is really interesting to us when I was showing you an example of fold switching. So RFA's NUSG family is a large family of universally conserved transcription factors. They are regulators, they bind to RNA DNA polymerase. They have a lot of members in their family, and in a paper which Lauren wrote in 2022, I believe, in Nature Communication, she showed how fold switching was pervasive in the family using computational prediction and really labeling the members of the family which switch fold and do not switch fold. And she followed up with experiments to verify those predictions. The point I'm trying to make is that, again, going back to this family, NUSG of this family usually exist in the C-terminal domain as a beta role, whereas RFAH, when it binds to DNA, RNA polymerase does undergo this switch, and 25% of proteins in this family, they do undergo the switch. But what we were interested in, and I'm going to show you a little data on this as well, because this is an ongoing project to really look at how fold switching occurs in this family. So far, we have seen example of evolved fold switching where the mutation had led to two different folds, but that doesn't occur in real time or at equilibrium as we saw for lymphotactin. Here, fold switching is triggered by binding to a partner, and it shows various members of the superfamily having fold switching, and it's pervasive. So there are different ways fold switching have evolved in these families, and they are fascinating to look at. So this is the tree that I want to talk about a little bit more. So here what we have done using the computational prediction method that was described in Lawrence's paper, we could label the members of the family as fold switchers or non-fold switchers, and we built a tree, and we have rooted them at this point because these are the oldest members of the family. We knew that for this particular family. What I want to talk about here, and what is really the point that I want to make here, is that when you look at the clades here, NUSG here, are mostly non-fold switching. And when you look at the fold switching that occurs here for RFHFS, you see that occurs here, but it occurs like everywhere else, also for the other members of the family. So this is particularly of interest to us because here you see fold switching happening back and forth. And to verify that, we have taken different variants of these families from everywhere on the tree, some from here, some from here, some from here. And we will be performing experiments to make sure that what we see in the phylogeny makes sense. So if that is true, this is a very unique way fold switching has evolved in this family, which means that it evolves at various points of the tree. And you can have a clade where you have both fold switchers and non-fold switchers, which is also very exciting. It is quite different from what we have seen from for lymphotacting as well as the bacterial response regulator. So again, to end the talk and to leave you with this message that fold switching is widespread, it is evolutionarily selected for and can be encoded by various different sequences. And it's important to understand how these occur and how evolution has selected for them in different families. And we see examples of that. With that, I would like to thank the organizers. Thank you for having me. I would like to thank my supervisor and all my lab mates and collaborators. Thank you. And I can answer any question that you may have.